All right. Well, welcome everyone. Welcome to session 2, room 4. This is our very much our hard science, one of our hard science sessions with quite a bit of work in the biology realm of things. I'm definitely not the one who's conversant. That's why we have our researchers here and our faculty mentors here. But I also know, after having many conversations with them, how hard they've worked on these projects. And I want you all to, celebrate along with us their hard work over this past summer, as well as I know in the fall have continued on it, and I know for a fact will continue work throughout the given academic year. All right, so what I've shared screen right now lists our presenters. We've got our first group here is Alexis Cox, Florence Mumby, and Ella Missy, and then followed will be Safik Villakanti. Am I butchering your last name, Vic?
No, you got it perfectly.
Okay, good. And then we'll follow up with our FSU iGEM team. There are too many to mention now, but I know Jovi, their team lead, will share their individual names as we come along. In addition, you see the QR code has our program. If you haven't searched it yet, I assume you have because you are here. But as a reminder and having access to it, in addition, after the event is over, later this week, I'll be taking the recordings of all the presentations, posting them onto our website as well as in the program. Obviously, you'll have to download a new version when we put all those recordings there, but it's also a nice keepsake, potentially digital keepsake for our presenters and our audience. I know that we've got some parents, proud parents in here as well. All right, let's make sure, I want to remind our speakers that let's keep an eye on our timekeeper, Natalie, so we know where we're at in terms of time. But without further ado, I want to invite Dr. Parvatiyar. I apologize, I should have asked the pronunciation at the beginning. And I was inviting you to share and introduce our first research team. You're muted at the moment. Are you able to unmute, Michelle? No? Okay. So should we have, I don't know if the other professor is here at the moment. Is Dr. Nagpal here? Are you able to speak to the students? All right, we must all be having some technical difficulties at the moment. So let me
now I can talk. Okay, I just couldn't talk before. Okay. Hi, my name is Michelle Parvatiyar, and I'm in the Nutrition and Integrative Physiology department. And I'm really pleased to introduce the 3 students who will be sharing their work with you. So Ella Missy is a biochemistry major who plans to go on to medical school. And then Alexis Cox is a senior biology major who intends to get her PhD in a cellular and molecular biology program and to do research that's related to inflammatory disorders. So very much in line with the work that they're going to show you. And then Florence Mumby is a biochemistry major who wants to go on to do an MD PhD program and has since joined my lab to finish up her undergraduate research. So back in the spring, I received an email from these students in search of a host lab to perform their project for this idea grant. And I was willing, and I brought Dr. Ravi Nagpal on board, who's an expert in the gut microbiome, and he's been incredibly helpful. And together, I think we've made a really excellent team. But overall, I've been really impressed with these students, their drive and commitment, and their ability to move this project forward and to learn all the techniques needed to get to the point where it is. And I would recruit each and every one of them to do a PhD with me if I could. So I'll turn over the floor to Alexis.
Okay, let me share the screen. Can you stop screen sharing? Yeah. Okay. Whoop. And Ella will be our first speaker.
Good evening, everyone. I will be presenting the first part of our project, which is entitled Superimposing Celiac Gut Microbiome Metabolites on Healthy CACO2 Cells to Observe Inflammatory Effects. For those of you who aren't familiar with it, celiac disease is a gastrointestinal autoimmune disorder which has become increasingly more prevalent in the population. The mechanism for this disorder is essentially that when someone who's afflicted with it, consumes gluten, which is a protein that is found in wheat, rye, and barley, the immune system recognizes it as a foreign agent and attacks it along with the epithelial intestinal cells. This results in the flattening of the villi or the cilia in the intestines. And the flattening of these villi can cause a multitude of unpleasant symptoms, most notably the inability of the patient to properly absorb nutrients. While celiac diseases become much more common in the population, there's still much to be learned about it. The gut microbiome describes the population and concentration of bacteria in the small intestine. These bacteria are essential for the metabolic pathways of digestion and other intestinal processes. The gut microbiome is different for every single person. It's essentially kind of like a fingerprint. More research has been done into the gut microbiome lately. The gut microbiome has a large effect on gastrointestinal health, but can also affect other areas of health, including and not limited to cardiovascular health, immune health, and even mental health. Celiac disease's inheritance pattern is still largely unknown. There's a gene that is associated with celiac disease, but there are people that have this gene that do not display pathology of the disorder. Because of this, there are a lot of people that think that celiac disease has a secondary effect or a different cause. Many people postulate that this is the gut microbiome, mostly because those with celiac disease in recent studies have been found to have different concentrations of bacteria in their gut microbiomes. Some of them have more of a certain type of bacteria and some have less of a certain kind of bacteria. Our research investigates this. We essentially aim to better understand the relationship between the gut microbiome and celiac disease and determine whether or not the differences in the gut microbiome between celiac patients and healthy individuals is a cause or an effect of the disorder.
So here's a brief outline of our experimental design. We had 5 healthy subjects and 5 celiac subjects, collected their fecal samples, created a fecal conditioned media, which just contained the gut microbiome metabolites, added these to our CACO2 cells, which are our intestinal cells, and ran qPCR for inflammatory markers. We chose CACO2 cells because they are a common model of intestinal cells in vitro. They are immortalized human colorectal adenocarcinoma cells. So they're basically cancer cells from the human colon. And we cultured them in glucose DMEM, 10% FBS, 1% penicillin and streptomycin, and L-glutamate. The larger picture, so the top picture on the side, is that when they're about 80 to 90% confluent, which is normally when we would split them, and then the bottom picture is when they're about 10 to 20% confluent, so they still need to grow a lot. Next, we created our fecal conditioned media. We took 30 milligrams of our fecal samples, added 10 milliliters of diamidum, vortexed it and shook it, then ran it through 3 different filters of decreasing size. The purpose of this was to extract not just the fecal matter, but also like intestinal cells that got into the fecal samples and bacterial cells from the gut microbiome to just leave the metabolites, such as short-chain fatty acids, acetate, propionate, butyrate, that sort of thing. We could not directly expose our cells to bacteria because that would kill our cells, and we want to measure inflammation, not cell death. So once we had our fecal conditioned media, we added the CACO2 cells to 6-well plates, and at 50% confluency, we added 100 microliters of our fecal conditioned media. After 24 hours exposure, we measured inflammation markers by extracting RNA, reverse transcribing to DNA, and running qPCR on tumor necrosis factor alpha, interleukin-6, interleukin-1 beta, interferon beta, MX1, and L32 as a control. Tumor necrosis factor alpha was chosen as it is a cytokine, and interleukin 6 was chosen because it's both a cytokine and an anti-inflammatory myokine. So it both signals that there's inflammation and helps fight off inflammation. Similarly, interleukin 1 beta is in the same class as interleukin 6, and interferon beta, and interferon is an inflammatory signal protein. So it's like a cytokine. And MX1 is a interferon-induced protein, so it's like a secondary inflammation marker. L32 was used as a control because it is just a ribosomal protein, which should not be affected by inflammation. Florence?
Weren't able to hear you, Florence. Speak one more time just to check. Does any of the other team members have her notes? So you can.
Do you want one of us to present her part? Or
Yes, please.
okay, I can wing it. Sorry, Florence. So here we have our first round of results, our first trial of results. As you can see, there is no, I'm just gonna quickly skim through this since we don't have time. Not really a significant difference in any of these. The red is the non-celiac, the blue is the celiac. And that there's a trend for inflammation markers to increase, but it's not statistically significant. To improve the statistics, we need to run more trials with a larger sample size and next time control for gluten-free versus non-gluten-free lifestyles, which we did not do. But the most important part is the skills we've learned along the way. Future directions include metagenomic sequencing, measuring short-chain fatty acid differences between groups, examining individual microbiome, the effect of individual short-chain fatty acids on cells, and creating a in vivo version. We would like to acknowledge Dr. Nagpal and Dr. Parvatiar for letting us use their labs and for just all the help they've given us along the way. Our generous fecal donors, because it is really hard, like it is not a fun process to donate poop. And our funding from the Helen Louise Lee Undergraduate Research Award and the 2021 Phi Eta Sigma Undergraduate Research Award. So thank you. Apologies for the technical difficulties. Sorry, I did not get to go over more of the results. We were running out of time.
Thank you for sharing nonetheless. And I know that you guys will be continuing to work hard on this project over time. Right now, do we have any questions? Please put them in the chat. Yes, I appreciate the encouragements as well. Great job. Dr. Tomko asks.
I can answer this one. For us to figure out whether or not the metabolite concentrations were similar, we would need to essentially kind of sequence all of the gut microbiomes from the fecal samples that we collected. This process is lengthy and expensive, so with the funding and the time that we were given, we didn't necessarily have time to do it. As a follow-up to the research, we've talked about having them sequenced just to see the different concentrations of bacteria. If we know what the concentrations of bacteria are, we can infer the metabolite differences, but we weren't able to do that in the first stage of our research. This was all very preliminary, so.
And I can answer Dr. Fadul's question. Yes, we would like to look more at sex differences, but because of our small sample size, and we only had, I think, one, because celiac disease is way more common in females than males, so we only had one male celiac sample. So that would be something to control for. It would be preferable if we could control for that. So there might be differences, especially I think it has been shown that there are differences between male and female gut microbiomes. So that does need to be looked at more.
Well, thank you very much. I appreciate your hard work and getting this into something that's also consumable for someone, for us lay people. All right. For our next presenter, Satvik Bilikanti, I'm going to invite Dr. Tomko to, you should have the ability to unmute yourself at this point, to introduce and tell us some kind words about Vic. Hopefully.
Yeah, so of course, it's my pleasure to introduce Vic Bilikanti. He's a junior here at FSU and is majoring in health management, policy, and information, which is a relatively new bachelor's program under the interdisciplinary medical sciences degree program here at the College of Medicine. Vic is a Florida native from the Orlando area. And in the future, he's looking to make the difficult decision between pursuing a medical degree and a PhD in some facet of biomedical research. So Vic actually originally joined my group as a UROP fellow, which is an FSU program for some of the brightest undergraduates coming in their freshman year. And his idea project is actually an extension of the work he started here as a UROP fellow. So hopefully you can help me in welcoming Vic here, who's going to tell you a little bit about some of the tools he's developing to investigate a promising drug target called the proteasome, which can be used potentially to treat some infections caused by a poorly understood family of parasites known as Microspiria. So Vic.
All right, I'm going to be sharing my screen now. All right. Thank you for that wonderful introduction, and I'm going to begin. So I would like to begin by thanking the Scott and Nina McNichols family, whose generous support provided me the wonderful research experience. So during, like Dr. Tomko said, during the summer, we looked at Microspiria. Microspiria are unusual obligate intracellular parasites. Microspiria are found to only live inside other cells of other organisms. Approximately 17 species of Microspiria can cause human infections, and if untreated, can cause damage or even death to immunocompromised patients. These species spread easily through contaminated water, and despite this, there are no reliable treatment options for microsporidial infections, and thus there is an urgent need to develop new drugs and to combat these parasites. On the right, you can see a Microspiria inside the host cell. We decided to focus on an unusual proteasome called the 26S, which regulates protein degradation for cell function. You can see on the panel on the right, there are several subunits missing in it compared to humans, such as those shown in green that are essential in all other organisms. And the subunits present in blue are often highly divergent of that of humans, which makes the inhibitors valuable for microsporidial drugs, which would be helpful in the future. Our goal is to create tools to manipulate, purify, and visualize these proteasomes from Microspiria, which would help us for drug screenings in the future. Also, Microspiria depend on M26S for normal cell health, and the inhibition blocks parasite replication in other animals. Unfortunately, there are no tools available to study the M26S. We use specific antibodies called nanobodies that come from camelids such as alpaca or camels. They are specifically derived from antibodies. They are a single polypeptide chain that can bind to a molecule of interest such as an antigen. In our case, it would be specific subunits on the proteasome. We use yeast surface display and have found and there's over 50 million 500 million individual yeast cells, each expressing a unique nanobody on its cell surface. On the middle panel, you can see how this is done, where each nanobody has each yeast cell has a distinct nanobody on this cell surface. On the right panel, you can see that the M26 is as bait. We can go fishing for yeast cells that produce a nanobody that tightly binds to the bait, and which can also wash away any unbound cells, and then either repeat the process or analyze the clones that bind. We first needed our bait, in which in our case, we used alpha-1. We could genetically introduce a functional handle called biotin onto the protein, which would help us immobilize the protein on our bait to be used when we go fishing for the right nanobodies. We also used E. helem because it is a species that infects humans but can readily be cultured in lab. We also chose alpha-1 also because we could produce the purified protein in high amounts of the common bacterial lab strain E. coli. By attaching a small molecule, like I said before, biotin to the alpha-1 protein, we could then immobilize it on magnetic beads as a handle for manipulation. On the right panel, you can see how this is done with the alpha-1 being highly expressed, which shows to be an effective or an attractive target to be used for further study. We could then use MAX, which is magnetic affinity cell sorting, to specifically isolate clones that bind to E. helem alpha-1. In the cartoon below, you can see how this is done. The nanobody is attached to the surface of a yeast cell, and any yeast cell expressing the nanobody will recognize the antigen and will stick to the magnet, and then can be isolated using a powerful magnet. The cells that don't bind to the antigen will fail to stick to the magnet and will also be washed away. Also, there can be non-specific binders where some cells can bind directly to the magnetic beads themselves, and these cells will be removed in the second experimental step. On the right panel, you can see this is an actual instrument that we've used in the lab, which has a strong magnet that can attract any nanobodies that bind with the antigen and wash away any that don't. To further enrich the positivity of the nanobody, we use FACS, which is fluorescence affinity cell sorting, which uses a fluorescent molecule to detect further positivity. We modified alpha-1 with a fluorescent molecule called M-cherry. We used a cell sorter to separate these cells that bound to M-cherry labeled alpha-1 cells and the ones that did not bind alpha-1. In the panel to the right, the cells that bound to alpha-1 would thus have a fluorescent ring that is attached to the cell surface, whereas the cells bound directly to the beads would not have fluorescence at all. On this panel on the right, you can see that each dot is a specific cell, and the ones on the bottom left are not fluorescent, while the ones on the bottom right are fluorescent. This specific panel shows the majority of cells in the naive library that make nanobodies, but that do not bind to M-cherry alpha-1, but not with M-cherry alone. This indicates that the cell is specifically binding to the E. helem alpha-1 part of the M-cherry alpha-1 fusion and not the M-cherry portion alone. This also shows that the cell is specifically recognizing and binding the E. helem alpha-1. This image is very interesting because it shows the specific nanobodies using FACS that can specifically find our antigen alpha-1. Each panel here on the left shows negativity or shows the M-cherry E. helem, but without the M-cherry or without the antigen binding to it. But on the right, you can see this is the M-cherry E. helem alpha-1 that correctly binds to the correct nanobody. Just like the bottom right, which we would need, we found 4 clones that have been identified that associate with the M-cherry E. alpha-1, but not M-cherry alone. This shows a very important milestone in our project. Finally, I would like to conclude by saying that we have had a successful enrichment of yeast clones expressing E. helem alpha-1 binding nanobodies. Like I said in the previous slide, we have found 4 positive cells, nanobodies, that can go through further testing to be potential cures for the parasite Microspiria. Our next steps are to clone and produce the recombinant nanobodies in E. coli and to test the most promising nanobody clones for ability to purify M26 from E. helem infected human cells. Finally, I would like to acknowledge once again the Scott and Ina McNichol's family for their amazing summer opportunity and the Tomco Lab for their time, and the IDEA Grant for giving me the opportunity to do research in the summer. Thank you, and now I am open for questions. So Dr. Tomko asked if it's possible that more than one of the clones you isolated are actually making the same nanobody. Actually, no, because the clones that are isolated are making individual specific nanobodies on the specific antigen, which would make it very unique for each cell. So just like that, we found 4 positive cells, and basically each nanobody is expressing a different antigen.
So I think David is having technical difficulties here.
The mute is off. Is it?
It
Yeah.
doesn't matter,
working?
but can you hear
There
me?
you go. We can hear you.
Now you can.
Don't touch anything.
Oh my God. Because my screen still says unmute on it. It's just,
Interesting.
I was talking for like 5 seconds and then nothing. All right, I'm touching nothing.
Don't blink. Yeah.
We always seem to crash Zoom somehow with these events. Yes. All right. Wonderful. Thank you for sharing, Vic. I know a lot of the students have got a lot of new knowledge and knowledge out of that, and also some of the innovative ways in which you are conducting research on campus with Dr. Tomko. Before anything else messes up, I'm going to hand the floor over to Caesar to introduce the FSU iGEM team, and then on to Jovi, where he'll sort of introduce the team, and then we have a video presentation. And Caesar, don't forget to hit that optimize for video when you go to share the screen.
Absolutely. All right. Well, welcome everyone. This is we've been part of the President Showcase, and I believe this is the 3rd year. This is the largest audience, so that's great. You know, one positive of doing this virtually. All right. So the iGEM stands for the International First of all, can everybody hear me? Okay. Beautiful. So it stands for International Genetically Engineered Machine. It's not our name. It's a name that was invented at MIT. It's a competition that started in the Boston area back in 2004, centered around MIT, but the first participants were 4 teams from the Boston area: Boston College, MIT, I believe Harvard. I forget exactly what all 4 were, but the bottom line was that it was just a local competition. And but now today in 2021, there are 355 teams with all continents represented worldwide as part of the competition. So it has grown dramatically in the last 15 years. So we, Florida State participated for the first time in this competition in 2017. And so this is the 5th year of iGEM here at FSU. And so it is a tremendous privilege for me to be with this team and like the teams before it, just an incredible group of very talented young human beings and emerging professionals. So it's truly a privilege for me to work with them. I'm going to take just one extra moment just to emphasize one bit about iGEM. Is that okay, David? Is the timing okay, or am I going over?
Yeah, we'll be fine.
Okay. So let me do a quick screen share here. I'm also doing the dual screen, so sometimes I pay the price for that. And hopefully we won't today. And so can everybody see my spreadsheet?
Nope. Oh, I can. Okay, good. Glad somebody
Beautiful. So these are the members of the team, and I just want to point towards the degree, the majors. So one of the missions of FSU iGEM, it's definitely about doing science, and in this case also doing engineering. but it's more than that. It's about doing team-based science and engineering. So one of them we have developing and maintaining excellent multidisciplinary teams. And I wanted to highlight that because that's one of the things that does distinguish iGEM from other research experiences on campus. And so when you do that, when you have a group like this one, where you have a biology major, an entrepreneurship major, a digital media production major, an environmental science major, how do you talk to each other? How do you work together? Turns out it's very hard. And actually, you're going to see the video that we put together, and you'll discover the challenge of having people between disciplines talk to each other about the work that was done. And we're learning. Now, this video we're putting together is actually a new requirement of iGEM. Before, we were doing a presentation just like Savik and our other young colleagues here today presented. We were doing a presentation just like everyone else. But iGEM in 2020 decided let's start making presentation videos instead of a slide deck. And so we iGEM, FSU iGEM, are still learning how to do that. And so you're gonna see some of the roughness that comes with that, but I'm confident, just like we've done in the past, that we'll learn how to do it well and then excel as as this team has done and the team before have done. So anyway, it's a great pleasure to introduce the 2021 FSU iGEM team. It is an amazing group of young students, and a real pleasure for you to see their work. So over to you, Jovi.
Hey, I'm Jovi Osagi, team lead of the 2021 FSU iGEM team, and it's a pleasure to present our project to you. You saw our team list earlier. I will be answering your questions as they come in, and I'll be delegating those questions to my teammates.
All right.
Yep.
So let's go ahead and then get to the video. How's the sound for everybody? Beautiful. So far, so good.
It doesn't matter where you are in the world or who you are. No matter what continent, religion, ethnicity, culture, generation, class you might belong to, we all wake up each day and face the same primal problem. Food. Food insecurity is defined as a lack of consistent access to enough safe, nutritious, and socially acceptable food for an active, healthy, and productive life. It exists on a spectrum ranging from uncertainty of where your next meal is coming from, to having no food at all, to having an abundance of non-nutritional foods. In 2020, over 700 million people were food insecure around the globe. In the US, over 35 million people are food insecure. The pandemic has exploded out food insecurity to 45 million people. Out of those 45 million, 15 million are children. Statistically, food insecurity hits the US communities unequally, as the rate of food insecure African American and Hispanic households is more than double the rate of white households. The Big Bend, home to the capital of Florida and FSU's main campus, has witnessed a boom in food insecurity from 14% to 13% during the COVID-19 pandemic. 1 in 3 children struggle with food insecurity every day. To learn more about the causes and effects of food insecurity in our community, we spoke with Second Harvest of the Big Bend, the largest food bank in the Big Bend area. Food insecurity in the Big Bend is primarily a socioeconomic issue. An estimated 15% of families in the Big Bend are working paycheck to paycheck. An economic downturn, a natural disaster, or a surprise expenditure is all it takes for a household to experience food insecurity. This results in trade-offs, forcing people to choose between spreading out their prescriptions to afford rent or avoid paying the utility bills their children can eat. A 2017 USDA report found that income is associated with 3 out of 10 chronic diseases, but food insecurity is associated with all 10. Communities where residents have very limited access to healthy and affordable foods, or food deserts, have high rates of food insecurity, and FSU's campus is surrounded by multiple food deserts. In an effort to combat the nutritional deficiencies in their community, local food pantries are asking for more fresh produce. However, fresh produce donations only last 1 to 2 days when they're received by the food pantry, leaving next to no time for the people who need it most to receive it, let alone use it. Food pantries are open anywhere from every day to once a month, making it hard to store fresh produce over long periods of time and reliably get it out to their clients. We are proposing to partner with PicoMart, a provider of debit card accessible refrigerators. The refrigerators filled with fresh produce would be located in communities experiencing food insecurity. Within the refrigerators, the fresh produce would be wrapped in Wrap, a chitin-based bioplastic that is biodegradable. The fresh produce would also be treated with Shield, a chitosan-based protective coating that extends the shelf life of the fresh produce. We expect the Fresh system to significantly increase the shelf life of fresh produce with the goal of supporting balanced meals to as many members of our community as possible. So what is chitin and chitosan? Chitin is the main structural materials of insects, shellfish, and fungi. It is produced by many species in various kingdoms for a variety of uses. Chitin is based on glucose, the central energy molecule used by life. It is a polymer of UDP, acetylglycine. Chitin synthase joins the acetylglycine together to form chitin. Chitin has preservative properties and can be made into a plastic material. Chitosan is a derivative of chitin. It has antimicrobial and antifungal properties while being safe to humans. Chitosan has an amine group on the second carbon of the glucose ring. So how do we make chitin inexpensive and sustainable? Chitin is sourced mainly from shellfish, which is expensive and unsustainable. Purification of chitin from these sources requires a highly alkaline solution that then needs to be neutralized. The Fresh system has the potential to increase shelf life of fresh produce on a large scale. We hypothesize that microbial production of chitin is easier to purify, more inexpensive, scalable, and sustainable. We designed, built, and are testing engineered E. coli intended to secrete chitin called the chitin secretion cells. We are converting brewers' spent yeast into an inexpensive growth medium and are testing it by growing the chitin secretion cells. In collaboration with the Lambert High School iGEM team in Sewanee, Georgia, we are testing an inexpensive lyophilizer to freeze-dry the chitin secretion cells for transport and long-term storage. In collaboration with the University of Pennsylvania iGEM team, we are characterizing 2 proteins identified in the UniProt database that may be chitin synthases. What about the chitin secretion cells? The chitin secretion cells contain the NOD-CIG generator, a genetic device designed to express the proteins NOD-C, NOD-I, and NOD-J. NOD-C is a chitin synthase that polymerizes UDP acetylglycosamine into chitin with 4 to 6 residues, which is known as chitologomer. NOD-I and NOD-J are chitologomer transporters that we expect will export the chitin outside the cell. The NOD-CIG generator was inserted into plasma backbone, and E. coli NEB5-alpha was transformed. We intend to also transform E. coli NEB-express. The chitin secretion cells are presently being tested. Proof Brewery provided us with brewer's spent yeast. Brewer's spent yeast is the yeast left over from the production of beer. We are evaluating if brewer's spent yeast can serve as the basis for an inexpensive growth medium for the chitin secretion cells. Testing is underway.
So David is still experiencing some technical difficulties. So this will be our Q&A portion. Hopefully you guys like the video. Yeah, so just ask us any questions that you have for our project.
So I'll add a bit that the actual presentation video, the presentation video that will be done at iGEM is at 20 minutes long. So there's more room there to tell the fuller story. Here we're trying to figure out how do you tell this bigger story in 8 minutes. And as you can see, we were challenged in doing it, and we're only partially successful. So we are learning. This is as much an experiment as as the project itself.
Can you hear me, Caesar?
I can.
Okay, good. I can't wait to look at that video on Slack because I couldn't see a thing.
Oh no.
But I know that all I'm going back to the celebration aspect of this. If I can't see the chat either, so if you see anything in there, please forward questions. But I know I'm very proud of this iGEM team, as all the past ones as well. But also thinking about Vic. Vic was in my Future Innovators class his freshman year, and he leveraged that to go on to Europe and now doing incredible stuff with Dr. Tomko. And I know that Florence, Ella, and Alexis should be very proud of themselves as well, even though they may feel like they've got a lot to do down the road. I know that the experience has strengthened them as researchers and bolstered their ability to take those next steps in life after they leave here at Florida State. Did we see any other questions?
Yeah, so we have questions from Dr. Tomko and questions from Ms. Foster or Dr. Foster. I'm not sure. Joe, you want to go ahead and
would it be easier if we just read them out, or?
Yeah, I'll read it out.
Okay, great.
So from Professor Tomko, do you have to feed the bacteria glucose in order to synthesize chitin? Or is the plan that they would make it from the spent brewer's yeast, which is presumably devoid of glucose since yeasts preferentially ferment? I will hand that question off to Chase, our implementation team member.
Hi, can you guys hear me? I can't actually share my video. That privilege has been denied. So
we can hear you, at least.
Yeah. So no, E. coli naturally I forgot the word, but they metabolize glucose automatically. So that part of the E. coli remains unchanged. And actually, brewer's spent yeast does contain some glucose within it, but it also contains a lot of other stuff that, like amino acids specifically, that then the cell no longer has to spend energy making that, and instead can use energy making our product.
Okay, next question is from Elizabeth Foster. How do you know when the experiment has worked? Do you test for food ripening or freshness? I will hand that question off to Jade Knight.
Hi, everyone. So we are currently working on this in our labs, this part of our experiment. But key factors we're looking for is shape change. So if it starts wrinkling or losing water loss, which is a big problem with tomatoes. And particularly, we are looking for fungal and mold growth, and in what percentage and at what rate it will be growing. We did take this from not necessarily fresh off the vine, because we're trying to replicate necessarily what people in, say, who are going to food banks, what type of food they would be getting. So it's not necessarily super fresh, super ripe, but definitely still in decent shape. And we are taking videos and photographs to make sure we have visual comparisons later on, which we'll be comparing. And as for the film, it would be more or less tested for its strength, because the chitin film is more for maintaining a certain environment, which would mean testing its permeability and its ability, which is pretty much if it holds water, releases water, holds gas, releases gas, as well as pH and temperature. So those would kind of be how we would test the freshness or ripeness of these vegetables.
Wonderful. Good explanation, Jade. Do we see
Joey,
anything else, Joby?
why don't you take on Dr. Tomko's first question? It's actually a good question.
Dr. Tomko's first question.
Yeah, the random question. Oh, that was a direct message. That's what I got.
Yeah, sorry, that one was for you. Just when they were talking about chitin, it made me think of something.
Okay, gotcha. I'll follow up with you then.
No problem.
Yeah, any more questions?
maybe I can just ask one more question. Is chitin breathable? Does it have good gas exchange? Do we know? I feel like it probably would.
Actually, it's the opposite. It's shown to be less permeable than polyethylene.
Really? How about that?
Yes.
I wouldn't have guessed. Very cool.
Oh, okay.
If that's it, thank you everyone for sharing. I'll take, as being someone who helps to supervise the program, I'll take the opportunity to say, if we have any students that are interested in joining the 2022 team for the IGEM program, we are meeting every single Friday at 3 p.m. in the pitch room of the Innovation Hub. We do combination info session, but as well as workshopping what kind of problem or design challenge, as I like to call it, the 2022 team wants to tackle. So we definitely invite all individuals, as Caesar showed, all majors to come have the conversation with us. And then with that, I'm going to wrap up. Thank you all for joining us. This concludes this session. We might, if you're interested, have one other session that's